Okay, welcome everybody. We are happy to be here and to be in this great room, in this great lecture hall. So we want to discuss now the reconceptualization of neuropsychiatric diseases with meta-analytical data mining. And as you can see here, that's the first picture of the Amadeo from 1553. So this nice building with this nice construction and architecture. We will have a flow of information. We start from introducing meta-analyses, what we can do with meta-analyses, what we can expect. The second talk will focus on psychiatric diseases, especially schizophrenia. The third talk will switch to neurological diseases, so to atypical Parkinsonian syndrome, and how we can predict symptoms individually in subjects. And the last talk will discuss how we can translate all these findings to single patient level, which is quite important, of course, so that we can also find a way to the single Amadeo and how to treat it. Okay, we will start with the first presentation by Veronika Müller.
All right, I will start with a more educational beginning and introduce you into neuroimaging meta-analysis, show you some examples of applications, and in particular, show you some studies in major depression. In the last 2 decades, neuroimaging has evolved into a widely used method to investigate brain structure and function, as well as the pathophysiology of brain disorders. Since the beginning of neuroimaging, the number of studies has steadily increased, with now more than 3,000 published fMRI studies every year. However, it has been shown that there are a lot of effects which are not replicable. For structural brain behavior associations, it has, for example, been shown that there is only little spatial overlap of results of independent studies. Similarly, for task fMRI, low overlap has been reported for thresholded statistical maps of the same contrasts. What the 2 studies both show is that this lack of overlap decreases with increasing sample sizes. In general, in single fMRI studies, they often suffer from low sample sizes and are therefore often underpowered, especially clinical studies. This, on the one hand, may lead to missed findings, but on the other hand, might push researchers to analysis approaches which may lead to increased false positives. Another factor that might lead to a lack of replicability might lie in the fact that the same mental process is often investigated with using different experiments, different approaches, and also analyzed in different ways. For example, interference processing is often investigated by using the color words group, but there are also a number of other versions of the tasks. Additionally, data can be analyzed in different ways by using different pre-processing pipelines, statistics, and software packages. A large study in 2020, for example, has demonstrated that analysis of the same data and hypotheses by 70 independent teams leads to high levels of disagreement. So insights from each individual neuroimaging study are limited by study-specific characteristics and effects due to low sample size, analytical and experimental flexibility, but also selective reporting and p-hacking should be mentioned here. It is therefore increasingly challenging to keep track of the wealth of findings and to distinguish spurious from replicable findings. Therefore, there is the need to quantitatively consolidate effects and to test what is consistently found. Until now, I only showed the problems of neuroimaging. However, there are a lot of studies And what they all have in common, despite their analytical flexibility, is that they are highly standardized, which means that they report their results as X, Y, Z coordinates in MNI or Tallarach space. And here, meta-analyses come into play. They synthesize the multitude of effects and test what is consistently found. There are two different approaches to neuroimaging meta-analysis. Image-based approaches base their analysis on raw data or full statistical images of findings, which has the drawback that it is often difficult to compile a data set which is representative of the literature. Coordinate-based approaches, on the other hand, are based on published coordinates of activation peaks, which has the drawback that they use a sparser representation of findings. However, as most studies report their results as coordinate, it has the ability to cover the full literature on a specific topic. In brief, coordinate-based meta-analysis extract coordinates from papers that investigate the topic of interest. Those coordinates are then taken. Their spatial uncertainty modeled by between template and between subject variance, the probabilities combined, and then tested against a null distribution of random spatial association between experiments, thresholded and corrected for multiple comparisons. With meta-analysis, we can therefore delineate robust networks that are associated to a specific mental process and consistently reported in the literature. We, for example, have identified a network which has been consistently found for tasks that use investigated face processing during different facets of empathy or impulsive behavior. However, we can not only test which regions are consistently found in a specific task or mental process, we can also see what is consistently found or reported when groups are compared, for example, a clinical population to healthy controls. The lab of Atkin did quite some work there, consolidating most of the psychiatric literature and showing in a transdiagnostic approach that there's robust convergence of findings across diagnostic groups, in grammata changes, but also in task activation alterations. We identified regions that are consistently associated to formal thought disorder and then used those regions for further functional connectivity analysis to compare patients with schizophrenia with healthy controls. And this is another interesting application of meta-analysis, to use regions that have been consistently associated to a specific mental process as priors and as informed networks for further analysis. What makes this approach so attractive is the direct link to a specific mental process, which makes interpretation often a bit easier. We, for example, used a lot of different meta-analytically derived networks involved in different tasks or in different processes, and calculated resting state functional connectivity within those networks for then to then classify patients or predict symptoms. For example, here we used resting state functional connectivity within different meta-analytical networks to classify patients with schizophrenia, here shown in green, or patients with Parkinson, shown in blue. And what we found is that different networks are differently informative for classification of schizophrenia or Parkinson, respectively. Additionally, we found that we could predict schizophrenia symptomatology by resting state functional connectivity within meta-analytical network, and in particular, cognitive symptoms based on functional connectivity within socio-affective networks. The remaining time of the talk, I now want to focus on major depression and how we can use meta-analysis to gain a better insight into the neurocorrelates of this disorder. In a first study, we wanted to investigate what has been consistently found and shown to show aberrations in brain activation in major depression during tasks of cognitive or emotional processing. Surprisingly, we didn't find any convergence at all, neither in the overall analysis across almost 100 experiments, nor in specific sub-analysis where we tried to analyze across more homogeneous samples and tasks. And when looking at the coordinates, we clearly can see that the results are scattered all over the brain. But what can we learn from this lack of convergence in neuroimaging meta-analysis? Importantly, this is not the only meta-analysis that didn't find any convergence. For example, inconsistent results have been found for insomnia disorder or ADHD. This can be due to a number of different factors, for example, uncorrected inference or publication biases, but most likely heterogeneity of the different studies plays a crucial role. So heterogeneity in the analysis approaches and the experimental designs, but with additionally doing group comparison, an additional layer of heterogeneity is introduced, and differences in investigated populations in comorbidity effect, medication effects might play a strong role. So on the one hand, the lack of convergence in clinical meta-analysis might point to problems in the original literature and to the need for more replication studies, but on the other hand, and what is potentially most likely in depression, it just demonstrates the clinical picture of a disease, which means that there are different alterations in this disorder, but there's no general pattern that we can find. We then try to reduce heterogeneity by reducing task heterogeneity and only focused on studies that used investigated gray matter volume in depression or resting state, and did another meta-analysis, and there we did find some convergence, especially in the subgenual cingulate cortex, left hippocampus, and amygdala, all regions that have been implicated in major depression. A recent meta-analysis that consolidated the findings of treatment effects did also find convergence in subcortical regions, but as well as in the insular and cingulate regions. So these three studies together in major depression show that for task activation studies, there are inconsistent findings pointing to a heterogeneous picture of this disease. But when combining across modality, things get a bit better, and we do find convergence. And for treatment effect, we find a network which show treatment effects, and there are also regions that are found in showing gray matter or resting state alterations which show treatment effects like the amygdala. For reducing the heterogeneity in our task activation data, we then, or dealing with the heterogeneity, we then focused on a network-based framework. So we were thinking that those regions that don't converge in our task activation studies might be or interacting with the same regions. So what we did, we used the coordinates we included in the task-based meta-analysis, so that showed alterations in task activation in major depression, calculated for each of these coordinates resting state functional connectivity using 100 participants of the Human Connectome project, combined those and tested this map for significance, and this map then reflects the voxel-wise integrity with brain regions that show task alterations during cognitive or emotion processing in major depression. And interestingly, we found that those sites are connected to specific networks. Here on the left is the brain regions which show connected to altered brain activation site in major depression during emotion processing, and on the right, the cognitive one. And we find highest integrities in regions implicated in major depression, for example, prefrontal regions and cingulate regions, including the subgenual cingulate cortex. And also, this network consists and includes regions that are found to show consistent structural and resting state changes, as well as treatment effects like the insula, the hippocampus, and cingulate regions. We then correlated our networks, so we did spatial correlation with previous models in major depression, and we found close similarities with other networks. So what we find here is that we don't find any localized convergence at all when just looking at it from a just localized perspective, but when trying to see with which regions those are connected, we do find a specific network, and targeting this network might then therefore affect all those regions that are reported, that have been reported to show task alterations in depression, which might also explain why, for example, stimulating the dorsolateral prefrontal cortex is effective in depression, even though this region is often not found to be dysregulated. So to take home, I hope I can convince you that meta-analysis is a powerful tool for delineation of robust networks, which can then be further used for analysis in, for example, clinical populations. The lack of convergence allows conclusions about the heterogeneity of individual studies, but could also demonstrate the clinical picture of a disease. Either way, they point to the need for more replication studies, pre-registration, and to follow best practice guidelines. From consolidating the results in major depression, we found consistent structural and functional alterations addressed in emotional control networks, which additionally show treatment effects, at least some of them, for example, the amygdala. For task activation studies, we do find inconsistent findings of aberrant task activations. However, when looking at them from a network perspective, we see that these distributed sites of brain alterations in major depression are connected to the same regions. And with that, I want to especially thank Simon Eickhoff, my group, and people from Düsseldorf and Jülich, all my collaborators, and you for your attention. And when you're interested in that topic, come and see those posters which are presented in the next few days at OHBM.
Okay, we have wonderful questions from the audience. I just select these ones which might be very important for application, and we cannot all, of course, answer. So one question is, can you comment on low-powered meta-analyses with liberal thresholds? Can we trust these meta-analyses? Maybe also what is the minimum number of studies which has to be involved?
Yeah, that is another thing that we found in, for example, the study of depression, where we didn't find any convergence at all. There are already a lot of meta-analyses that did find some converging results, but a lot of them were underpowered. And what has been shown is, for example, especially for coordinate-based meta-analysis, that it is important to include at least a minimum number of studies, about 20, that you can make sure that they are not driven by one or two studies, because it doesn't say anything if effects, it's not consistent, doesn't reflect any consistency anymore when out of 20 studies, 2 drive a result.
Okay. And second question is that meta-analysis is a useful tool, but one issue is the bias towards areas of the brain that register better, for instance, the brainstem, and that we have a bias there.
In general, we always, so with meta-analysis, there is always the problem of bias that we might have, also publication bias and stuff. But with regard, for example, for publication bias, we do test for spatial convergence, and therefore adding null results wouldn't change the effects at all. Also, we do have a bias by excluding region of interest analysis, but we would additionally induce bias if we would include them. With better registration or less, I am not sure how it would really affect the results of meta-analysis, because it wouldn't be found in all of the individual studies. So I think that should be noise that doesn't show up as convergence.
Okay, thank you. Thanks for your questions and your interest. And of course, the presenter, Veronika. Now we move forward to Su Lui. He will discuss meta-analytic imaging findings in schizophrenia and data-driven approaches to subtype schizophrenia patients. Because he is from China, he couldn't visit the conference, so we will have a virtual talk.
Good afternoon, everyone. My name is Zhu Lu from West China Hospital of Sichuan University. Today, my topic is about the meta-analytic imaging findings in schizophrenia and the data-driven approach to subtype schizophrenia patients. I have no conflict of interest to declare. As we know, schizophrenia is a chronic severe mental disorder with high relapse, high suicide, and high disability. Patients cannot tell what's real from what's imagined. And we know genetic and environment factors involved in the schizophrenia, but the etiology of the schizophrenia remains unknown. Because of that, there is lack of objective biomarkers for clinical work. As a famous psychiatrist said, psychiatrists remain the only medical specialist that never look at their organ they treat. For the last 20 to 30 years, radiologists have been searching for the biomarkers of schizophrenia using various kinds of imaging methods, including structure and functional methods, especially using MRI. Here, I want to summarize the meta-analytic findings of structure and functional MRI, especially in high-risk subjects, first episode schizophrenia, and chronic patients. For convenience, I mapped the findings of previous meta-analyses on the glass brain map. As shown in this picture, the blue dot means the decreased matters, while the red one means an increased parameters. As shown in these pictures, the studies of the relatives of schizophrenia exhibit either increased or decreased gray matter volume in various of brain regions, especially the decreased gray matter volume, especially in the prefrontal temporal regions, but increased volume in the basal ganglion area. When we look at the gray matter changes in first episode schizophrenia, they also showed widespread gray matter changes involving the basal ganglion, thalamus, and the cortical circuit. Here, the first episode schizophrenia is defined as the earliest duration is less than 2 years. And these findings is consistent with the changes in the relatives of schizophrenia, but they are more wide and more severe. However, when we take a look at the chronic schizophrenia patients, they showed more decreased gray matter volume involving the prefrontal temporal and the basal ganglion areas. Here, the chronic patients is defined as the mean duration of illness more than 5 years. When we put the figures together, we can get two messages. First, the changes of schizophrenia along the illness of the course is progressive, which is support the neuroprogressive hypothesis of schizophrenia. Second, we found the consistent change in the cortical, striatal, and thalamic loop, which might be promising quantitative imaging markers for schizophrenia. When we move to the functional MRI study of schizophrenia, the findings is different from structure studies. And shown in this picture, the high-risk people show increased activation in the right dorsal anterior prefrontal cortex, which is the most consistent findings. And the other findings varied across the different studies. When we look at the first episode of schizophrenia, they demonstrated decreased activation in the bilateral parietal and the medial prefrontal cortex, while increased activation in the bilateral putamen, superior temporal gyrus, and occipital gyrus. Especially the decreased activation of the medial prefrontal cortex is the most consistent findings in first episode schizophrenia. It is interesting to look at the change of chronic schizophrenia, which also demonstrated widespread increased and decreased function involving different brain regions, especially the decreased activation in bilateral postcentral gyrus, left precuneus and right occipital gyrus, while increased activations in the bilateral inferior frontal gyrus, bilateral superior frontal gyrus, left amygdala, left inferior temporal gyrus, and right anterior cingulate cortex and insula. When we put these functional findings together, we also can see a progressive change from the relatives to first episode schizophrenia and chronic patients. However, these findings are more widespread than structural findings and involving the default mode network, sensor mode network, visual network, and the silence network. Thus, the functional findings may be more state-related. Taking the structure and the functional findings together, the gray matter changes have been most robust within the thalamocortical network, while the altered brain activation have been most pronounced in frontal, parietal, and default mode network. As we summarized in our previous review, these findings indicate that the regional anatomic and functional brain abnormalities are significantly dissociated during the early course of the schizophrenia. One possible explanation is that the functional MRI may reflect the physiological changes related to the state, while the changes in brain anatomy reflect more stable and long-standing alterations. Taken together, there are complex cerebral abnormalities in schizophrenia. For structural studies, they found consistent findings within the biochemical cascade for schizophrenia, and the structure markers may be more suitable for subtyping the patients. While for functional studies, they found more widespread changes across the whole brain, which suggests they may be state-related findings of schizophrenia. But the relationship between the structure and functional findings remains unclear. The main challenging of the previous study is the inconsistent findings across different studies. Even in the meta-analyses, the small samples and several clinically relevant heterogeneity, including the genetic, the clinical symptoms, the cause of illness, and the treatment outcome, may confound the findings. One of the solutions to solve the problem is the subgroup the patients based on the clinic information or using data-driven approach.
Oops.
Many previous studies subgroup the patients based on the clinical symptoms. For example, Our previous studies compared the patients with prominent negative symptoms with those without prominent negative symptoms, and the findings showing that the patients with prominent negative symptoms show more widespread gradient changes. However, such type of studies based on the hypothesis that the patients with similar clinical symptoms have similar gradient changes. However, that is not true. In 2015, our group first used clustering analysis to study the white matter features of the schizophrenia. In the large sample of first episode schizophrenia, we found there are two different subgroups with different white matter changes. However, they showed quite similar clinical symptoms, except the group 1 with widespread white matter changes showed more severe negative symptoms. These findings suggest the possibility that similar clinical symptoms may have different green changes. Following that study, there are many studies using clustering analysis to subgroup the patients. For examples, we found 3 different subgroups of patients defined by the distinct patterns of regional cortical and subcortical morphological features. A similar 3 subgroup patterns was also identified in the independent data sets of patients from a multi-site DSNP studies. The patients showed quite similar clinical symptoms, but with quite different gradient changes. For examples, the group 1 showed quite severe gradient deficits, while the group 3 showed increased gradient volumes. However, these patients showed different cognitive changes. The patients with more severe gradient changes showed more severe Beck's scores. These findings suggest that the patients with similar clinical changes may have quite different gradient changes. Similar findings were also identified in non-institutionalized patients with schizophrenia, and those with more pronounced subcortical changes showed more deficits of cognitive function, but with similar changes of the clinical symptoms with the other subgroups. To explore the possible pathogenesis of these imaging subgroups, we also used data-driven approach to subtype the patients based on the other features of the patients. For examples, we explore the inflammation status of the patients, and we subtype the patients based on the DNA methylation and the T receptors. And the interesting findings is we found 2 subgroups of the patients using data-driven approach with these methylation and the T receptor features. And we found that there are 2 subgroups. The groups with the abnormalities of the immune system showed more severe gradient changes, and this is quite consistent with the group 1 type, the type with severe cognitive deficits. Most of the previous clustering analyses are using the structural features. For functional MRI study, the clustering analysis is still at the exploratory stage, where further validation and increased sample size are needed. However, these are quite difficult because functional MRI to date, its low signal-to-noise ratio and elevated monitoring cost make it a poor clinical alternative for studies. And if we want to using fMRI to study the subgroups of patients, a multi-site data collection with strong effort are needed in the future. However, the initial study using functional MRI also found different subgroups within the schizophrenia patients. For example, based on the 8 cognitive dimensions, using fMRI, they found 4 subgroups, and the group 4 showed more severe brain deficits of the frontal parietal regions, showed more severe working memory deficit. Another study using functional MRI also showed distinct patterns of neural recruitment during working memory. Taken together, data-driven approaches may be used to remove the heterogeneity of the imaging study of the schizophrenia, and the structural MRI-based subtyping study is more reliable than functional study, while the functional study is at the initial stage. Finally, I want to thank all the group members and our cooperators for the study. Thank you for your attention, and welcome to China and welcome to Chengdu. Chengdu is a nice city with the name of the paradise and also is the hometown of Panda.
So we thank Su Lui for his presentation. Interesting presentation about structural and functional combined meta-analysis in schizophrenia. And for the sake of time, and because of course we cannot answer the question related to his talk, we would switch to the next presentation by Franziska Albrecht, Disentangling Atypical Parkinsonian Syndromes and Predicting Symptoms Individually.
Thank you, Matthias, and I thank the committee for selecting our symposium and giving me the opportunity to present our work. So today I'm going to talk about how we use meta-analysis to disentangle atypical Parkinsonian syndromes. Parkinson's disease is just not one disease, it's comprising an entity, or it's just one entity of a huge group of Parkinsonian syndromes. And those Parkinsonian syndromes, they are similar in clinical features, but they have different underlying histopathologies. And up to now, to definitely discriminate Parkinsonian syndromes, histopathology is necessary. So, as I said, there are several Parkinsonian syndromes, but those Parkinsonian syndromes are also characterized by, on the one side, the clinical syndrome, so the clinical representation, and an underlying histopathology. So the typical representation of Parkinsonian syndrome is the idiopathic Parkinson's disease. This is a so-called synucleinopathy and described by Lewy bodies in the histopathology. Furthermore, there are several atypical Parkinsonian syndromes, but today in this talk I will focus on only 2, the progressive supranuclear palsy and corticobasal syndrome. Both have in common that they are so-called tauopathies and are described by different patterns of astrocytic tau inclusions. So since the OHBM is a bit more technical, I would like to guide you now through those diseases. Corticobasal syndrome is a really rare, so-called orphan disease. There's just one person per 100,000 individuals that suffers from this disease. It is characterized by so-called rigidity, muscle stiffness, bradykinesia, dystonia, so abnormal posture of hands or feet, a cortical sensory deficit, apraxia, so an inability to initiate movement. But furthermore, it is also described by a really unique syndrome, the so-called alien and anarchic limb phenomenon. This is really specific to this disease, but 30% of participants or patients with corticobasal syndrome exhibit this, where participants or patients feel that their arm or even also their legs have a life of their own. Another atypical Parkinsonian syndrome and also a tauopathy is the so-called progressive supranuclear palsy. This is also a really rare disease where 3 to 6 individuals per 100,000 individuals suffer from this disease. It is also characterized by several motor symptoms, but especially by the so-called progressive vertical gaze palsy, by early postural instability, which leads to falls backwards, and by also quite early executive dysfunction. This disease is described by a very well-known but not yet systematically validated pathognomonic marker, so a disease-specific marker. This is the midbrain atrophy. On the picture below, you can see on the, for you, left-hand side, brain image from a T1 image from a healthy participant, and in the close-up, the midbrain, so the structure that is mostly affected in progressive supranuclear palsy. On the right-hand side, you can see an example of a patient with midbrain atrophy in the close-up on the right side. There you can see that the midbrain gets thinner. So the issue is what is still missing are validated objective neuroimaging biomarkers for those Parkinsonian syndromes to disentangle them and also to support the diagnosis. What our working hypothesis is that we can identify, using meta-analysis, disease-specific atrophy that disentangles those diseases using MRI, the so-called Parkinson's disease, progressive supranuclear palsy, and corticobasal syndrome. So the general workflow is that there are single patient studies, so especially in those rare diseases, it usually starts with peculiar case studies where just individual diseases are represented. This is quite high in individualization, but low in statistical power. If there are more patients, then this can be summarized into a group study, where the common differences among a cohort are identified, which is then a bit lower in individualization, but higher in statistical power. But our approach is then to use meta-analysis, and as Veronica explained to us, is that this is how to identify convergence among small cohort studies, for example. So in our case, we used meta-analysis to identify disease-specific patterns in Parkinsonian syndromes. Our first approach was to use this in Parkinson's disease. Here we included multimodal studies, saying we included studies, 50 studies that used structural MRI, so gray matter atrophy, so to say, and 14 studies that used FDG PET, so glucose positron emission tomography. On the right-hand side, you can see the results in the upper row. You see hypometabolism in 2,323 participants with Parkinson's disease. Here we found that the areas of the inferior parietal cortex and the chordae nucleus were affected consistently among those patients. And in the bottom row, you can see structural MRI. Here we found that the medial-occipital cortex is affected, saying participants have lower gray matter in this area. But what we can conclude from this meta-analysis is that, in general, we found consistent functional abnormalities, especially in significant glucose hypometabolism, whereas structural MRI seems to be not so good to identify Parkinson's disease, since these were rather focal and rather inconsistent. But Parkinson's disease is also described by a huge clinical heterogeneity, which can be then more or less summarized in clinical subtypes. So we also divided our meta-analysis into those clinical subtypes. And here again, FDG PET seems to be more sensitive to identify differences between patients and healthy controls and patients with specific clinical subtypes. So for example, in the blue frame now, the clinical subtype of Parkinson's disease with cognitive impairment. Seems to be more impaired in cortical associative parietal regions. On the right-hand side, now in the blue frame, is Parkinson's disease with no cognitive impairment, so pure motor impairment. And interestingly, here we found the area of the caudate nucleus to be specific for Parkinson's disease with mainly motor symptoms. So we further used also meta-analysis to disentangle clinical syndromes from the underlying histopathology. In this case, we used corticobasal syndrome, which is the clinical representation, so the clinical symptom that you can see. But the underlying histopathology is called corticobasal degeneration, which can be only proven by histopathology. So here we included 11 studies, whereas several of those participants had a proven histopathology. So we divided our studies into clinical studies and into the cohort with proven histopathology. The overlap between those two cohorts could have been what was found in the pre-supplementary motor area and posterior mid-cingulate frontomedian cortex. And what we can conclude from this is that those overlapping regions are specific to the combination of the clinical representation of corticobasal syndrome with underlying histopathology of corticobasal degeneration. In the next approach, we used meta-analysis to validate the so well-known pathognomonic marker, but this was not yet systematically validated. Here we included structural MRI studies that investigated gray and white matter. Interestingly, we found convergence among those studies in the areas of cerebral and cerebellar peduncle, as well as the midbrain. Sorry. In the image below, you can see on the left-hand side the results of the meta-analysis, so the color for cluster, overlaid on a healthy brain. In the middle of this picture, you can see an example of a patient, and in the purple frame, the midbrain. And on the right-hand side, you can see the so well-known pathognomonic marker of midbrain atrophy, which is also known as the so-called hummingbird sign. Hummingbird sign, because of the thinning of the midbrain, it looks like a beak of a hummingbird. So to further validate this well-known pathognomic marker of midbrain atrophy, we did not just look at coordinate-based meta-analysis, we now looked at region of interest effect size meta-analysis. So here we compared, or we included studies that compared different midbrain metrics. On the right-hand side, you can see again the midbrain, and then a close-up, the midbrain, and here different measures, so diameters or the area or ratios. We found 15 studies that measured different metrics of the midbrain to identify progressive supranuclear palsy and to disentangle this from different Parkinsonian syndromes. And we could show with our study that in general, midbrain metrics distinguish progressive supranuclear palsy from other Parkinsonian syndromes with quite strong effect sizes. But in general, what we could find is that midbrain area, so the measure of the midbrain area, showed the largest effect size to distinguish PSP, progressive supranuclear palsy, from Parkinson's disease. So the question that you could ask now is this now support to incorporate MRI or this biomarker into revised diagnostic criteria? And to answer this question, We went further and looked into the literature, which other meta-analyses on Parkinsonian syndromes and also on other neurodegenerative diseases are out there and which areas they found, so which brain areas are affected in these diseases. And since this is quite a busy slide, it should be just seen as an overview, but I would like to draw your attention to the study that I showed you now, so progressive supranuclear palsy. There have been also other previous meta-analyses. All of them found the areas of thalamus, midbrain, insula, cerebella, and cerebral peduncle, as well as the caudate nucleus affected. But if you now go column for column, you can see that the midbrain is mainly affected in progressive supranuclear palsy, as well as in multiple system atrophy. But here, one should note that this is only in the red nucleus, so a specific part of the midbrain. Further, if you move on to the next column, you can see that cerebella and cerebral peduncle is only affected in progressive supranuclear palsy. So thus, it is more or less we could conclude that these areas are really specific to progressive supranuclear palsy. What I would like you to take home from my talk today is that Parkinson's disease is mainly affected in glucose hypometabolism, which seems to be more sensitive than structural atrophy. But it is not unique to the disease. So the patterns that we found can be also found in other neurodegenerative diseases. Progressive supranuclear palsy is described by midbrain and cerebral and cerebellar peduncle atrophy, which we would conclude is disease-specific and could be also included into revised diagnostic criteria as a bioimaging marker. Corticobasal syndrome is described by pre-supplementary motor area and posterior mid-cingulate frontomedian cortex atrophy. Here we could show that those areas are specific to the combination of the clinical representation of corticobasal syndrome with the underlying histopathology of corticobasal degeneration. And I would like to end my talk by thanking all my colleagues and collaborators at the Max Planck Institute and also at the Karolinska Institute.
Yeah, thanks a lot for the presentation. We have now a critical question, which is, of course, very good. The disease-defining hallmark of all Parkinsonian syndromes is atrophy of the substantia nigra, causing dopaminergic deficit. And the question is why the meta-analyses did not show such effects in the substantia nigra. And the critical point is then that if we cannot identify in the meta-analysis this histology, whether these meta-analyses are trustful. That's the point. So the question is whether it's sensitive or what might be issues.
Yeah. Okay, that's an interesting question. So in Parkinson's disease, what you can see with, at least with structural MRI, that there is a huge variation between studies that show no results at all. So we also had many null results. And the more the disease progresses, the heavier the atrophy. Let me think now. Maybe, okay, also the thing is, of course, there's a lot of variation between those studies, which we should regress out with meta-analysis. Good question, actually. Maybe it's because we don't see yet in the structural MRI. It's the substantia nigra.
Could it be? Is my micro working? Yeah. Could it be that you had a lot of region of interest analysis in there, and therefore they weren't included in the meta-analysis?
Also, good point. I mean, there are many region of interest analysis that don't look at all at the rest of the brain. And with the whole brain meta-analysis, there is a huge amount of null studies, right? since there is a huge variation depending on the disease progression. So in early disease stages, you don't see that much atrophy.
Another option is, of course, that histology is not reflected in MRI. I mean, not a one-to-one relationship. So, of course, you cannot detect very small areas, and you don't see histopathology. You just see volumetric changes or changes in chromatin density, and this might not reflect the changes in the substantia nigra.
Yeah, I mean, most of them were 3 Tesla. So.
We now move on with the next talk due to time limits, and Matthias will present his work.
Okay, now we have the last talk in the symposium. I want to tell you something about steps to go to single subject analysis, or to support single subjects with meta-analysis. So firstly, I want to introduce the most important point in clinic. The most important point in clinic is not statistics, it's not high number of subjects, it's this patient, the single patient. That's why I want to introduce a single patient for you. Then I want to discuss how we can increase evidence with meta-analysis. I mean, this is a point already discussed also in the other talks or addressed. Then I want to show you how we can develop concepts with meta-analysis. And the last point is how we can translate meta-analysis into clinical routine. So the first point is the individual patient in clinical settings. So this is, the example is a 51-year-old male real estate lawyer. He is at work and he spends most of the day at working, shuffling papers, reading magazines, or downloading pornography. Instead of working, he also embezzles money at work. When asked about his future, he is very optimistic, making lots of money, what real estate lawyers normally do, I would say, especially also in Germany. He loses interest in his wife and children. He watches TV instead of speaking and develops a strong desire for potato chips. And finally, his manners deteriorate. He eats with hands, stuffs his mouth. So we have deep changes in personality and behavior in this patient. By the way, the picture is not, of course, not the patient, but it's Friedrich Nietzsche from Leipzig, who was born nearby and also buried nearby Leipzig, the philosopher. And he was also discussed to suffer from frontotemporal dementia. The disease is so-called behavioral variant frontotemporal dementia. And as I have shown you, the typical symptoms are disinhibition, apathy, loss of sympathy or empathy, hyperorality, and also changes in eating behaviors. In diet and neuropsychologically, this disease is characterized by executive deficits and also by social cognition deficits. So we have now in the new classification systems, these are the international diagnostic criteria for BV-FTD, we have for new degenerative diseases several steps or stages. Possible is the clinical criteria are fulfilled, and probable means that imaging criteria support the disease diagnosis. In this case, there is frontal and anteriotemporal changes discussed to be specific for the disease. So how we can increase evidence with these meta-analyses? As already discussed, we have a lot of papers published. You see here in light blue, brain functional MRI studies published on Medline, and in dark blue, the dementia MRI studies. So we have a lot exponential increase, and we can use these great tools which are available even online and in the literature. So we can go, by going this way with the meta-analyses, we can go from case reports to group studies, to multicentric studies, to meta-analyses, and increase, of course, statistical power, we can generate generalizability. So now I want to discuss how we can develop concepts with meta-analysis. This is now focused on behavioral variant frontotemporal dementia, what I have shown you before, the patient. These are the results of our first meta-analysis. I think it was the first also which used anatomical estimate meta-analysis. And you can see here in the frontomedian cortex and in the anterior insula, there are the hot spots of the disease. So it's not the temporal region, how it was described in the diagnostic criteria, but the frontal areas which are mainly involved. And if we discuss the disease, whether the pattern, the imaging pattern is specific for the disease, we have to, of course, to compare it with other neurodegenerative diseases. And as you can see here, it is specific, so there is no overlap with the regions for Alzheimer's or primary progressive aphasia. These are other kinds of frontotemporal lobar degeneration beside BV-FTD. Why is the disease so interesting? I mean, first of all, it's people suffer from this disease, so the patients are not aware of the symptoms, and the relatives and spouses suffer a lot from this disease. So, of course, it's interesting to understand the disease and to treat the disease, but it's also a very interesting disease because regions are affected which make us human, so especially the frontal lobe, which increased its relative size across evolution. And one important point in the disease are von Economo-neurons. These von Economo-neurons are located mainly in the ACC and also in the anterior insula, and they are discussed to be related to socio-emotional functions and are found in apes, humans, whales, so in organisms which are highly developed. In behavioral variant frontotemporal dementia, we see a specific decrease in the density of these neurons in these areas. So this is a nice coincidence between histopathological data and the imaging data, what we obtained in the patients. However, I have discussed that we have a lot of publications on dementia and MRI, but we have a lot of brain functional MRI studies in healthy subjects. And the next point I want to discuss is how we can combine these data sets. So we can combine imaging data from disease with imaging data from healthy subjects. Here we have conducted a rather complicated meta-analysis. So in the first step, we wanted to identify the neurocorrelates of the disease, again, now separately for MRI, reflecting atrophy, structural MRI, and FDG PET, reflecting changes in glucose metabolism. And as you can see here, in red, you see the atrophic regions. It's again focused on the frontomedian cortex, anterior insula, and additionally the basal ganglia. And in blue, you can see changes in metabolism, in glucose metabolism, so these are hypometabolic areas. So we can validate the imaging diagnostic criteria and focus these to the frontal region. In the next step, we wanted to extract behavioral correlates. So for this purpose, we went to the probabilistic functional brain atlas of the brain map database. This atlas contained, to this date, 40,000 subjects, healthy subjects, and now 100,000, I think. And these are healthy subjects. So in this database, there were functional imaging studies of healthy subjects. And these are categorized according to several cognitive domains. And we calculated so-called behavioral domain profiles. Here we used the regions identified in the first analysis, where we identified the neurocorrelates of BV-FTD, and we looked whether in these regions we have higher numbers and activations in healthy subjects for specific cognitive domains. So we extracted the behavioral, psychological, or mental correlates of networks involved in BV-FTD. This database contains 5 cognitive dimensions and 83 paradigm classes. So this is a data-driven approach because we use another cohort, a healthy cohort with functional imaging data to extract the cognitive deficits in BV-FTD. These are the results. In blue, you can see FDG PET areas, and on the other side, on the right side in red, you see MRI regions, so which show atrophy. I just want to focus on two regions. Here, for instance, for the inferior frontal junction area, which is particularly involved, as known from the literature, with executive dysfunction, cognition was a significant finding for the functional study, which makes sense, of course, in this cohort of BV-FTD. And for atrophic areas, I just want to discuss this area, frontal-median cortex. There we had a lot of emotion, perception, and action. So this region is especially related to these functions. Additionally, we found other cognitive functions which are related to this network. So this was emotion processing, 5 clusters, reward, 5 clusters, cognition inhibition, 2 clusters, and pain processing. So we know also that these patients have a change in pain processing. They have a decrease in pain, actually. What is the limitation in this database in the BrainMap.org that, like in all other meta-analytic databases, you have a predefinition of the categories, so have a predefinition of which tasks are involved. And some tasks are not involved and are not reflected in the database, and so also empathy and theory of mind was to this date not reflected in the database, and that's why we have in a third step, we have combined the imaging meta-analysis where we identified the neurocorrelates of BV-FTD, here again shown in blue and in red, with the neurocorrelates in healthy subjects for empathy and theory of mind. And as you can see here in the yellow conjunction focus or region, you see that the frontal-median cortex seems to be a very important area where these functions interact with atrophy or changes in metabolism. And for theory of mind here in white as well, in the frontal-median cortex, there is an overlap so that these meta-analyses indicate that the frontal-median cortex is a hot structure in BV-FTD that is involved. Another interesting point is that BV-FTD persons might show changes in behavior which are related to moral cognition. That's why it was of interest also to look and to do a conjunction analysis for moral cognition, as shown here in, it's not really yellow, it's rather green. So moral cognition, and these are the networks which are involved in moral cognition in healthy subjects. Here again you see the areas which are affected by BV-FTD, and again we have an overlap here, as shown in yellow in this case, in the frontal-median cortex. So this is also a very important region why patients might be impaired in moral cognition. And of course, what are the consequences if you are impaired in moral cognition? You might show criminal behavior, and this is indeed the case. So in behavioral variant frontotemporal dementia, approximately 40% of the subjects show criminal behavior, like sexual advances, theft, public urination, and violence. This is an important issue because sometimes you don't know that the person changes, elderly person, and commits such criminal behavior, and you don't know why they are doing that. So this is the concept we actually developed. I cannot explain the details. I only just want to show that the frontal-median structures are the hotspot for the disease, and which is a hub which connects all these atrophy changes, hypometabolism changes, and cognitive changes in the disease. And based on these different cognitive functions, we can also reconstruct the symptoms, what they show, like loss of empathy, mind blindness, or dysexecutive syndrome, disinhibition, and apathy. Okay, so we can, with these approaches, we can reconceptualize diseases, not only BV-FTD, but also other diseases, of course. How can we translate meta-analysis into clinical routine? So this is the next point I want to discuss. How can we bring the results of the meta-analysis back to the society, how we can improve treatment. So although we increase a lot the generalizability with the meta-analysis, the important point is we lose personalization. So we lose the single patient. The higher the numbers involved in studies are, the less the single patient is reflected. And we have to go back and return to this area, to the patient. So we have used these meta-analyses, the results of the meta-analysis for BV-FTD, to optimize detection of BV-FTD. These are results for support vector machine classification to identify patients in comparison to controls, and we reach across the whole brain an accuracy of 82%. But if we go to the ROIs extracted in the meta-analysis, we can increase the accuracy, so we can utilize the meta-analysis to improve diagnosis of the patient. At the end, I want to present another patient, but in this case, it's a 50-year-old woman with bilateral stroke lesions in the so-called inferior frontal junction area. This happened in the territory of the middle cerebral artery due to atrial fibrillation, quite common disease, of course. She complained cognitive slowing, impairments in memory calculation, word finding difficulties, so a rather blurry pattern of symptoms. These are the MRI images. You can see here, these are the lesions, and they are exactly in the inferior frontal junction area, which is discussed, as I already mentioned, to be connected to executive dysfunction. We looked for the neuropsychological test results. You can find the pattern here. It's rather complex, so we used a lot of different neuropsychological tests, but most important are executive functions. These are percentile ranks compared to healthy controls, and here you see a very strong impairment, especially in executive functions, which is very interesting because the inferior frontal junction is with this model, the patient as a model, you can validate that this is a good theory to think that the inferior frontal junction is explicitly related to executive dysfunction and function, of course, in healthy people. So this is like a PROCAR, like a case report like PROCAR has done or Wernicke, but we wanted to do something else. So don't be afraid that Mars attacks start now. It's just that I want to introduce another method. That's meta-analytical reading of symptoms, called Mars, where we can predict the symptoms in single patient with meta-analytical tools. So here you can see, again, the lesions of the patient. So the single patient, what I have presented before, in the left and in the right, inferior frontal junction area. This was, of course, connected with a larger network involving other regions. And these are the results for the behavioral domain profiles. So we went again back to the brain map database and extracted the cognitive functions which are associated with these brain regions, and these are the results. And what you can see here on the right side, it's Stroop test, working memory, so classical executive function tasks. And on the left side, there are a lot more tasks, but also NBAC, so again, executive function tests, but also language tests, or language paradigms which show activation in normal subjects there. And interestingly, this patient indeed did show a language impairment in the beginning, right after the stroke, but later on the language improved, the language functions, and the executive deficits remained. Okay, I want to now summarize the results of the talk, or what I've presented here. So I think that meta-analytical data mining can reveal neural networks facilitating diagnosis and differential diagnosis, that we can use it to reconceptualize neuropsychiatric diseases, that we can also use meta-analysis to support personalization in medicine if we combine it with other methods like machine learning. And on this way, from diagnosis of neurodegeneration to therapy inducing neuroregeneration, which is, of course, an ambitious, very ambitious aim, we can use a magic black prediction box, which also other talks have discussed. And in the end, I think we can predict diagnosis, differential diagnosis, symptoms, histopathology, if we additionally involve, of course, other methods like PET, amyloid PET, tau PET. We can also intend to use personalized therapy approaches, and we can also predict prognosis. Thanks a lot for your attention, and if there are any questions, please ask these questions.
So the highest ranked question for you is, since von Economo neurons are mainly in layer 4 of ACC and anterior insula, have you looked at ultra-high field fMRI?
On what? On?
Ultra-high field.
No. Unfortunately, actually, of course, I didn't systematically look for studies in this field. I'm not aware of studies, 7T or something like that, with BV FTD, but it would be a very interesting study, of course, to use 7T MRI to look specifically for changes in the frontal-median cortex in BV FTD. But I don't know systematically the literature, actually. But to date of the meta-analysis, there hadn't been any studies with 7T, unfortunately, of course.
Okay, then one last question. When are we ready to admit that for some disorders there are simply no effects?
So, yeah. This was a very good question in my mind. Have we to admit sometime that we don't have effects in some diseases? So I remember a talk by another person who spoke about schizophrenia and GWAS analysis. And then he reported the results of the GWAS analysis with schizophrenia, and then he had a GWAS study with 50,000 persons suffering from schizophrenia, and there hadn't been any consistent effects. So, okay, 50,000 subjects with schizophrenia, no effects. One might admit that maybe it's not relevant, the GWAS study for schizophrenia, and we don't have effects. But the presenter told that we have to increase sample sizes to find effects in GWAS analysis. I mean, of course, it's an important point. When can we stop to collecting subjects to admit that there are no effects also in our meta-analytical studies? I think it's clinical relevance, for instance. If I would say that 50,000 subjects are not enough in GWAS studies for schizophrenia to find effects, and I don't see effects, then I would say, oh my God, it's clinically not at all relevant. Yeah? And I mean, the same is the case with meta-analyses, of course. But as all of you know, absence of evidence is not evidence of absence. So I mean, even if we do not find effects, it might be the case that we do not have enough numbers, but I think the point is to discuss relevance, yeah? How high the numbers of subjects have to be involved in studies. And if I go to the view of a patient, I mean, it's also good if you don't find effects. I mean, it's good if you don't find in meta-analyses atrophy effects. For instance, in Parkinson's disease, the atrophy effects are not that strong, and that's good for the patient, for instance. I mean, it's bad for the scientist because he thinks he cannot publish it, but nevertheless, you can also publish the negative findings to avoid publication bias. Thank you, and thank you for your that you visited our symposium, and we hope you have a great time in Glasgow.